Good afternoon, everyone. We are so happy that you're here. Thank you so much for joining us. We have a record number of participants, which is really exciting. I'm Monica Coenraads. I am the founder and the CEO of the Rett Syndrome Research Trust. And my inspiration and my motivation come from my daughter, Chelsea, who has endured with an amazing amount of patience, dignity, and strength everything that Rett syndrome has thrown at her over the last almost 30 years. Despite seeing her daily struggles, I am filled with optimism. I've seen firsthand the Rett field go from obscurity to a high-profile disorder, and a disorder that the biopharma industry is increasingly more and more interested in. I really want my optimism to be contagious today, and I hope that all of you are going to leave this webinar uplifted. Joining me today are some of my amazing colleagues, but I'm going to introduce them as they present. Please go ahead and put your questions in the Q&A at any time during the webinar, but we'll address them at the end. Okay, so let's dig in. Okay, so most of you know that the Rett Syndrome Research Trust is focused exclusively on genetic medicines. But what does that really mean? It means that we're targeting the MECP2 gene or the RNA by either supplementing or by correcting the mutations. Now, because the mutations in MECP2 impact so many systems in the body, and because so many drugs have been tested in the animal models of Rett with little success, we chose years ago to focus on genetic approaches. We felt that doing that provides us with the best opportunity of having the kind of dramatic improvements we want to see as parents, as well as being the best use of RSRT's time and resources. Now, genetic medicines are made up of two components: the cargo, which is what we are trying to deliver into the brain. So for gene supplementation, for example, the cargo is the MECP2 gene. For base editing, the cargo is the editing machinery. Now, you can have the best cargo in the world, but if you can't get it into a sufficient number of brain cells, it's just not going to be able to do much. So the other equally important component is the delivery technology, which means how you get that cargo into brain cells. And it's that combination of cargo and delivery technology that makes up a genetic medicine. Now, it's important to understand how a genetic medicine goes from a discovery in an academic lab all the way to a commercialized product that patients can access. Now, most discoveries happen in an academic lab at a university. And as a side comment, I'll mention that historically, this is where RSRT has focused, funding to academic labs. But as the science has matured, that has changed, with our focus and funding now mostly lying with biotech firms. Now, once an academic lab discovers something worth protecting, the team files for a patent, essentially putting a legal fence around the idea so no one else can copy it. In step 3, the patent gets licensed, meaning a company gets the rights to take it from the lab and to develop it further. Step 4 is enabling studies. Now, this is where the science gets stress tested. Researchers run experiments to prove the idea is safe and actually works well enough to move forward. Scientists are typically looking to answer questions like, does the drug harm animals at certain doses? How does the drug behave in the body? Can the medicine be manufactured consistently and at sufficient quantity and quality? Essentially, you're building that evidence package that convinces regulators that it's safe enough to move from animals to humans. With the data in hand, the team moves on to step 5 and begins talking to regulators. For example, the FDA in the US or the EMA in Europe, and so on. Think of this as an ongoing conversation to make sure everyone agrees on the path forward. If those conversations go well, regulators give the green light. And in step 6, you get official approval to start human trials. Now, in the US, there are some steps that have to happen in between receiving approval to start a trial and actually dosing the first patient. You have to receive approval from the ethics committee, called an IRB, at each of the clinical sites where the trials are happening. And you have to negotiate a budget with each of the participating clinics. As you might imagine, genetic medicine trials are very expensive. Step 7 is where the medicine is tested in real people in carefully controlled studies. If the results are strong, the team goes back to the regulators in step 8 to seek full regulatory approval of the product. And finally, step 7, commercialization. That's the finish line. That's where the product launches and becomes available to patients. And in preparation for this, companies will have been talking to payers, insurance companies, governments at the state level in the US, national levels in Europe and beyond, to ensure that they will pay for the product. Now, as you might imagine, there are challenges to overcome with each of these steps. Developing genetic medicines is not for the faint of heart. Now, several years ago, RSRT announced our strategic plan, Roadmap to Cures. It's our goal to have submitted applications to begin clinical trials for 3 novel genetic medicines to regulatory authorities by 2028. Now, some of you might be asking, why 3? Well, one of our philosophies at RSRT is to have multiple irons in the fire at all times. We never want to be caught in a situation where a program doesn't go forward for whatever reason, and we've got to start from scratch. Those of you who were around in 2021 will remember when Novartis terminated the Rett gene therapy program that got its start at RSRT. And while that was very disappointing, it would have been a devastating blow had we not had other irons in the fire. And we can't predict which approach will deliver the best results. So we pursue multiple approaches, more shots on goal, more chances of dramatic improvement. We follow the data, we double down on what's working, and we prune what doesn't. And there is nothing magical about the number 3. If we have the financial resources to advance more programs and the data looks good, great, we'll advance more. So who is RSRT? Before we dig into the genetic updates, I want to give you a glimpse into who we are and what we do day in and day out. We have in-house, full-time, fully dedicated staff that has decades of industry genetic medicine experience, as well as clinical and regulatory experience. You'll have an opportunity to meet Bob Deans and Jenny Laforet, and you will see for yourselves what a resource that they are. Beyond the industry experience, we have Rett experience. Now, that may sound obvious to many of you. Of course you have Rett experience. You're a Rett nonprofit. Yes, we are. But we have John Sinnamon, who you will meet in a few minutes, who worked hands-on at the bench, deeply immersed in everything MECP2 for a decade. He knows the Rett literature inside and out, and he's a great asset, not just to RSRT and to our community, but to the companies that come to us often for advice on Rett programs, on their Rett programs. And I don't know of any other Rett nonprofit that has a John equivalent. So if I had to explain what RSRT does on a daily basis, I would say the following. We're recruiters. We're always on the lookout for academic scientists, companies, startups that are working on relevant technologies. We do our best to encourage them to apply their technologies to Rett. We're funders. Money always helps grease the wheels. We don't do requests for proposals. We did that for many years at my first nonprofit. And I think that was appropriate when we were trying to fertilize the field. But at RSRT, we decide internally the areas we want to be active in. We identify the scientists and the companies that we want to work with, and we figure out a way to make that happen. We're marketers of Rett. And this was especially important when Rett was still at a relatively unknown disorder. Fortunately, we've come a long way. We are conveners, whether that's through our consortiums or our genetic medicine symposiums. We bring people together with a goal of accelerating timelines. We're brokers. This has become a critical aspect of what we do. As the science has matured and the technologies being pursued have increased, we make connections between academics and industry programs, again, to accelerate timelines. And we're risk takers. We're not scared to invest time, energy, and money in areas that other funding agencies, perhaps more conservative ones, would likely take a pass on. We're patient-centric. This is a word that I often hear in industry, and I know the companies mean it when they say it, but they have challenges that we don't have. They have investors and shareholders and Wall Street analysts that they have to make happy. Our shareholders are you, the community, and especially our donors and our fundraisers. We answer to you, and you want what we want. We want to make our kids dramatically better. So here's our research team. You're going to meet Bob, John, and Jenny in a minute, but we're incredibly lucky to also be working with Dave Backer. Dave has an adult daughter with Rett syndrome, but professionally, he works in the CDMO industry, which manufactures viral vectors. So he's been a founder and a CEO, and he volunteers his time to RSRT and is playing a critical role with regards to our viral vector manufacturing efforts, which you're going to hear more about. We also have Adrian. Adrian Bird was one of the first scientists that I reached out to 28 years ago when Chelsea was diagnosed. Adrian has had an outsized impact on the Rett field. He discovered MECP2 in the early 90s. He made the first animal models. He showed us that Rett-like symptoms in mice are dramatically reversible. And he's contributed enormously to understanding what MECP2 does. When I decided to start RSRT, Adrian was the first person I went to. And I asked him whether he'd join our board of trustees. He said yes. And 18 years later, he's still on board today. He's provided his perspective on every single project that RSRT has funded during those 18 years. Rounding out our staff and Adrian are advisors with legal and business development expertise, program management experience, and genetic medicine consultants. It's a really impressive group that augments and strengthens our internal team. Our only non-research colleagues are Tim Freeman and Stacy Rizal, who is our administrative officer, and Tim is our chief development officer. Tim has a teenage daughter with Rett, and Stacy is the proud aunt of Bryn, who also has Rett. And I think it says a tremendous amount about RSRT and what our focus is when you look at the ratio of research to non-research staff. Now, because the best predictor of future behavior is to look at past behavior, let's review some of RSRT's accomplishments. This will be quick, and then we're going to jump right into our genetic medicine updates. So I mentioned previously Adrian's reversal experiment. That experiment was funded by RSRT's predecessor, the Rett Syndrome Research Foundation, where I was a research director. I'm including it here because that reversal experiment changed everything for me. It was the impetus to start RSRT and for taking the decision to focus on genetic medicines. Our funding to Adrian and others has contributed greatly to understanding the MECP2 function. We funded the first gene therapy experiments. This goes way back, starting at 2010. And I think it's worth saying that we championed genetic medicines for Rett before it was fashionable to do so. It's easy to jump on the bandwagon when genetic medicines are the obvious choice, but it's a heck of a lot harder to do when it's the unconventional thing to do. We funded the first RNA editing experiments. We launched the Gene Therapy Consortium. We funded the mini-gene development. We funded work to develop MI-Rare and Exact that are now in the Taysha and Neurogene programs. Oh, sorry about that. Yep, okay. We licensed CapX capsid. You're going to hear more about that soon. We're funding non-viral projects. We created a patient cell line biorepository, and that has been such an enormous resource to the community. We've had 102 unique requests for those cell lines. They've been sent out to 34 companies and 66 academic labs in 16 countries. We funded dozens of mouse models, and we launched a few years ago the Rett Syndrome Global Registry, which is an online platform open to any Rett family around the world where you can share key information about your loved one with Rett. The registry is available in 10 languages, and if you haven't joined yet, please do, The information you possess about your child is so valuable. It's too valuable not to share it and to put it to good use. So please click on the link in the chat. We've also established a digital natural history study with the technology company Citizen Health. This is available for now to US families. With caregiver permission, Citizen extracts data from your child's medical records to increase our body of knowledge about the natural history of Rett and to provide a benchmark for what better looks like if we had effective treatments in genetic medicines. Findings from the first group of participants was recently presented by our Chief Medical Officer, Jenny Laforet, at the annual meeting of the American Society of Gene and Cell Therapy, which is the premier genetic medicine meeting in the world. We're about to submit this work for publication in a peer-reviewed scientific journal. For both the registry and the digital natural history study, the more people who take part, the more robust and accurate the body of data becomes. It's a powerful way to get involved and for you to have a big impact. So please make sure your child is represented. Now, we've done all of this and more while allocating an average of 92% of every dollar donated to our research program. In summary, I'm proud to say that RSRT has played a key role in putting Rett syndrome on the genetic medicine map. We simply would not be as far along as we are without RSRT. Okay, so now that I've provided some groundwork, our Chief Medical Officer, Dr. Jenny Laforet, is going to pick things up from here. Jenny is a physician scientist with 20 years of experience in biotech and pharma, focused specifically on rare genetic diseases. She has led gene therapy programs for Angelman syndrome, Canavan disease, Duchenne muscular dystrophy, and she brings all that experience to Rett. Welcome, Jenny.
Thank you so much, Monica, for the kind introduction, and hello, everyone. So as Monica introduced, as you can see, we certainly set an ambitious goal for ourselves to get 3 programs cleared to enter clinical trials by 2028. But fortunately, there has never been a better time to move genetic research into the clinic, as there's really been an explosion of interest in applying scientific advances to therapeutic development for Rett. The field has now matured to the point that multiple genetic approaches are progressing through animal studies with an eye towards being clinic ready in the next 2 years. RSRT has been supporting a broad array of genetic medicine strategies that we'll review momentarily. But before we dive in, to put some context around these different genetic medicine approaches, I'll start with some background about genes, about DNA, RNA, and proteins. The title of this slide is The Central Dogma, which sounds pretty grandiose, but it's appropriate since these processes underlie pretty much all of biology, in bacteria to humans. I'll start with proteins because they are literally the stuff we're made of. Proteins are large molecules that have 2 main functions in the body. Some proteins make up the material our body is composed of, like skin, muscles, inner organs. Other proteins run the multitude of functions needed to make our bodies work. MECP2, the protein that is abnormal or absent in Rett, is the second type. It controls expression of other important genes in the brain. So how are proteins made? Almost every type of cell carries the instructions for making all the proteins in the body. And these instructions are in the form of long linear molecules called DNA. The DNA instructions for each specific protein are called genes. When there is an error in the DNA instructions, this is called a gene mutation. There can be missing bits, added bits, wrong information, even a single letter that's wrong, and other kinds of mutations that can prevent MECP2 from being made properly. In order to use the information in the gene to make a protein, a copy of the DNA instructions is made for the cell's protein production machinery to read. This copy is called messenger RNA or mRNA because it carries the information to where it's used. If there's an error in the gene, the messenger RNA will contain the same error. So based on how the system works, you can imagine there are multiple steps along the way where an adjustment or a correction could be made to overcome the effects of the mutated gene. So this slide now shows the kinds of genetic medicine approaches that RSRT is supporting. So genetic medicine approaches. To orient you to the slide, the list on the left in blue shows the methods that focus on the mutant DNA sequence in the MECP2 gene itself. And on the right in pink are the approaches designed to fix the errors in the messenger RNA that's copied from the mutant gene and that provides the actual instructions for making MECP2. So starting on the upper left with the DNA-targeted programs. The program that's furthest along is gene supplementation, in which cells are provided with a working copy of the gene of interest, in this case MECP2, to take over for the non-functional gene. The next approach in the list is called base editing. You'll be hearing more about this from Bob later, but briefly, this is a technique that can swap out a single mutated letter in the gene with the correct letter, so the instructions for making MECP2 are now correct. It's analogous to fixing a typo in a document using the cut and paste function. You may have heard about baby KJ. This is the approach that was used to treat him by correcting a single letter in an important gene that controls metabolism in the liver. So prime editing operates by similar principles to base editing, but it allows substitution of a string of letters, not just one. It too is a very promising approach, but it's being backburnered for now as prime editing in brain cells is still quite challenging. And finally, on the DNA side, MECP2 reactivation is a different kind of therapeutic strategy that, as the name implies, aims to restore MECP2 function by reactivating the healthy but dormant second copy of the MECP2 gene on the X chromosome that's been silenced in approximately 50% of cells. The science behind this method is less far along than the other, so it too is being backburnered at the moment. So now switching to the RNA side on the right, RNA editing is just what it sounds like, namely correcting the single letter typo in the messenger RNA that was copied from the mistaken information in the gene. For longer RNA stretches that need correcting, trans-splicing uses a targeted pairing mechanism between two strands of RNA in which the correct sequence is swapped in to replace the mutant one. You'll be hearing a lot more about gene supplementation, base and RNA editing, and trans-splicing from John and Bob later in the talk. So I would like to make one further comment about the two approaches that we're putting on the back burner for now. We feel that now still both technologies need to make some more progress before we can invest further. As it happens, we did the same thing with trans-splicing a few years back, but in the ensuing time, scientific progress was indeed made, and now we've brought trans-splicing back, full speed ahead. So now let's turn to more specifics about gene supplementation. As mentioned, gene supplementation delivers copies of the healthy gene to compensate for the original mutated non-functional gene copies in cells. The healthy gene is delivered to cells using a non-disease causing virus called adeno-associated virus, or AAV. Now, AAV-mediated gene supplementation has a track record of success already and has already been used in several approved gene therapies for a variety of disorders, including spinal muscular atrophy, hemophilia, and Duchenne muscular dystrophy. And this is also the approach being pursued by Neurogene and Taysha in their Rett gene therapy programs, which we'll be talking about in an upcoming slide. So AAV was selected as the delivery vehicle because it doesn't cause illness, it can't reproduce in the body on its own, and it causes less strong immune reactions than viruses that were used previously. The MO of most viruses is to attach to cells and deliver their own genes to hijack the cell and trick it into making more virus. In the case of AAV gene therapy, and we can look at the next slide for this, for a diagram, we manufacture a so-called recombinant version of AAV in the lab by giving cells that are grown in a flask 3 sets of instructions. First, the genes needed to build the AAV outer shell, or capsid, and to grow more virus. Second, to fill the capsid, instead of the native AAV genes, we provide the gene of interest, in this case MECP2, that includes additional sequences that will let it be packaged into the AAV. And then third, some helper genes are included that support production of the recombinant AAV. So at the end of the process, instead of a fully functional AAV, we've made a recombinant AAV that, as shown in an earlier slide, serves as the delivery truck that goes to the intended address. It's the capsid that attaches to targeted cell types and then delivers the cargo, the MUCP2 gene that it's carrying. So the advantage of AAV gene supplementation is that it delivers a healthy copy of the MUCP2 gene, no matter what the original mutation was. So in principle, any patient with a non-functional MUCP2 gene could be a candidate for this approach. But there is a major limitation to keep in mind, though. Based on the current state of the science, AAV can only be given once. Though milder than other viruses, AAV still causes an immune response in the body. So if the immune system were to see AAV again in the future, it would recognize it and mount a so-called memory response. This is actually how vaccines work. The immune system remembers what it's seen before and leaps into action more vigorously the second time. And this is great if you want to stop an infection that causes illness. But in the case of repeat AAV dosing for therapeutic purposes, it's a problem. So the immune system will clear the second dose of AAV from the body, so it can't get to its target cells and do its job. In addition, the memory response is stronger than the original response and can actually cause collateral damage to tissues, making redosing risky from a safety standpoint. So now let's turn to the Taysha and Neurogene gene supplementation programs that are currently in clinical trials for Rett. And these are outlined on this slide, side by side. There are many common elements between the Neurogene and Taysha programs. Both gene supplementation approaches use a well-established type of AAV called AAV9 and inject a quadrillion AAV particles into the cerebrospinal fluid, or CSF, the fluid that bathes the brain and the spinal cord. But there also are some differences. First, the route of administration, how it's given into the body, is different. The Neurogene product is injected through a single burr hole that's drilled into the skull and then injected with a needle that goes through the brain tissue to reach one of the lateral ventricles. The lateral ventricles are spaces that are filled with the CSF, the cerebrospinal fluid, and they occur on both sides of the brain, and that circulates throughout the CSF space. So this sounds scary, but it's a common procedure and it's generally well tolerated. In contrast, the Taysha program injects into the CSF via a lumbar puncture or spinal tap between vertebrae, bones in the lower back. And then also the cargoes differ. Neurogene includes the full-length MUCP2 gene, along with some elements that regulate expression of the gene, whereas the Taysha product uses an abbreviated version of the MUCP2 gene that includes just the key functional parts of MUCP2 to allow space for its own set of regulatory sequence to all fit into the AAV particle. So both programs are now in the so-called pivotal stage when the final definitive clinical study is being conducted with the goal of submitting the data to FDA for regulatory approval. Both these trials are now fully enrolled, and the companies plan to share data later this year and early next year. So thank you. Over to you, Monica.
Great. Thank you so much, Jenny. Okay, so now I'd like to introduce you to a newcomer to the Rett syndrome gene supplementation space, a company called Reverta. Reverta is a startup biotech firm from Milan, Italy. Rett syndrome is their one and only program, which means it receives their undivided attention. The scientific work behind their program was done by Dr. Vanya Broccoli at Ospedale San Raffaele in Milan, a world-renowned institution for genetic medicines. Reverta is delivering a full MUCP2 gene using CapX as the delivery vehicle. Many of you will remember that last year, RSRT announced that we had negotiated a non-exclusive license to CapX, a viral delivery technology that is expected to cross from the bloodstream into the brain. I'm going to ask our Director of Research, Dr. John Sinnamon, to tell you about the cargo and the delivery aspects of the Reverta program, as well as explain some of our other genetic medicine approaches as well. John is a molecular neurobiologist who has spent much of his career working on Rett syndrome. He did his postdoc work in Gail Mandel's lab, where he helped pioneer RNA editing for Rett. He was an active member of both of our gene therapy and MECP2 consortiums, and more recently, he spent 3 years at Pfizer, leading a team developing messenger RNA and genomic medicines. He joined RSRT full-time a year ago, and he's helped RSRT significantly up our game. Welcome, John.
Thank you, Monica, for that kind introduction. As we mentioned, Reverta is a startup company that's developing a novel gene supplementation design, and they're combining next-generation delivery with the capsid CapX and a cargo that uniquely addresses 2 obstacles for MECP2 and Rett syndrome. As you may have heard before, MECP2 has been described as a Goldilocks gene, where too little functional MECP2 results in Rett syndrome, and too much MECP2 expression causes MECP2 duplication syndrome. Achieving this balance is most difficult in cells that are not affected by MECP2 mutations, because those cells already have the correct amount of MECP2. Female patients, which are very common in the Rett syndrome field, most commonly have approximately 50% of cells that are unaffected. They're mosaic because MECP2 is expressed from the X chromosome. The second challenge is that MECP2 is not expressed in every cell at the same level. For instance, neurons in the brain express much higher levels than non-neuronal cells. These are not easy challenges to overcome, but that is exactly what Reverta's cassette is designed to do, while also reaching brain-wide distribution by combining this technology with the CapX capsid. So what is CapX and how does it reach so many cells in the brain? Most therapies are largely blocked from reaching the brain because of the blood-brain barrier, or the BBB, which evolved to keep the brain safe. AAV9, which is currently in the clinic for Rett syndrome and other neurological disorders, has a limited ability to cross this barrier when introduced intravenously. To overcome this limitation, groups including Tatia Neurogene deliver AAV9 into the cerebral spinal fluid, or CSF. This is the fluid that bathes the brain and the spinal cord, but it also flows in 4 chambers called ventricles within the brain. By introducing the virus into the CSF, it allows passage into the brain by bypassing the BBB. But importantly, it doesn't allow penetration into all brain structures because they all don't have access to the CSF. In contrast, the brain is very highly vascularized. Almost every cell is touching or extremely close to blood vessels. And newly engineered capsids that cross the blood-brain barrier allow for intravenous delivery of the therapy and a lower dose, which increases safety. So how is all of this accomplished? Well, CapX binds to a well-studied receptor called the transferrin receptor, or the TFR1 receptor. And that receptor shuttles iron across the BBB and into the brain. CapX uses that same mechanism to hitch a ride on the TFR1 receptor and distribute itself through blood vessels throughout the brain. Now, importantly, TFR1 has been well studied and used as a druggable target in the past. CapX, on the other hand, is relatively new and has not yet been tested in human patients. The goal for first-in-human trials for other disorders using CapX are expected to start within the next year. So this is a great demonstration of what I was discussing. On the left-hand side is an adult mouse where AAV9 encoding a green fluorescent protein was delivered into the CSF. And you can see that there are regions of the brain that become green. And when you zoom in, you can see some of the individual cells that light up. This approach is important because it's relatively safe. It's been in thousands of patients, and it was the primary method for reaching the brain for a very long time. On the right-hand side is that same dose of virus and the same cargo, but it's delivered using the recently developed CapX. Now, clearly there are more cells that are expressing the green protein throughout the brain. And we also know from all of the preclinical work that the more cells that we can reach with Rett therapies, the greater the improvements are expected to be. So what is our role as we start this collaboration with Reverta? As we've discussed before, RSRT has a non-exclusive license to use CapX for Rett therapies. One of the most important aspects of making any drug is actually the manufacturing. Obviously, the amount and purity of virus that's being dosed into a mouse is not the same as what's going to be put into human patients. And as you increase the scale, there's always unforeseen difficulties. RSRT is committed to doing the unglamorous but necessary work of understanding the engineering challenges that come with making sufficient doses of CapX for clinical use. And we've allocated $1.1 million to do just that with Reverta and their new technology. Equally importantly, we're working in concert with the Reverta team, contributing to their joint steering committee and contributing our unique expertise to help with experimental design and their regulatory path to help them to achieve their goal of submitting for regulatory approval in 2028. So the next few approaches that I'm going to talk about relate to repairing MECP2 mutations. Now, by repairing mutations, whether they're at the RNA level or the DNA level, you can avoid these issues of overexpression and the cell type differences I mentioned, because you're not adding any more MECP2 than what is already in the cell. So the first therapeutic modality is very near to my heart, as it's something that I worked on very early on in my career as a postdoc, and it was how I was introduced to RSRT and Monica and this wonderful community, and that's RNA editing. So RNA editing refers to a process where enzymes that are already present in our cells, called ADAR, change individual bases in an RNA molecule. Now, this ability has been harnessed for Rett syndrome therapeutics, and they use a guide that acts as a GPS to direct the ADAR enzymes from their normal targets, additionally adding MECP2 to one of their target lists. And this allows MECP2 RNA to be repaired. Now, this guide tells them not only to go to MECP2, but which individual base to repair. So this means that for a subset of Rett mutations, we can actually repair the mutation at the RNA level. Now, the mutations that are amenable to this approach are R168X, R255X, R270X, R294X, G to A mutations, and C terminal deletions. Now, the guide RNA is unique for every mutation because it's a GPS and the mutations are in different places on the RNA. But they can be delivered by virus, like CapX, or using a small chemically modified piece of RNA called an antisense oligonucleotide, or ASO. Now, ASOs are small enough to be able to distribute into the brain when delivered into the CSF, but they do need to be dosed several times a year because they're degraded over time, unlike viruses. Now, RSRT has an ongoing collaboration with a company, ProCure, that is developing ASO guides to repair patients with the mutation R270X. In the future, other mutations can be addressed in what is referred to as an umbrella trial. RSRT is committed up to $3.7 million to develop RNA editing therapies with ProCure. We also provide our scientific and regulatory expertise as they move into the clinic, with a timeline for their regulatory submittal in 2027 with patients for R270X. But importantly, while that program is progressing, they're also pursuing the other mutations that can follow in this umbrella trial. Next slide, please. While ADAR-mediated RNA editing makes single base repairs to MECP2 RNA, trans-splicing uses the cell's natural machinery to replace large sections of RNA. So an analogy would be that RNA editing corrects individual typos, whereas trans-splicing replaces whole words. Now, the advantage is that instead of developing individual therapies for each mutation, all of the Rett-causing mutations can be addressed with just 2 therapeutics. Unlike RNA editing, trans-splicing does require just viral delivery. You don't have the choice between viral or non-viral with ASOs, but can use a capsid like CapX. You go to the next slide, please. RSRT is working closely with Tacit Therapeutics to help develop this technology. Using an initial funding award of $1 million, Tacit is using their AI-assisted high-throughput screening to identify trans-splicing molecules that can efficiently repair MECP2. Now, together with our help, their goal is to submit a program for Rett syndrome for regulatory approval in 2029. And now I'm actually going to transition over to Bob Deans, our CTO.
Yeah, thank you, John. That was great. So now it's my pleasure to ask Dr. Bob Deans, our Chief Technology Officer, to present our base editing program. Bob's resume speaks for itself. He's a serial founder and company builder in cell and gene therapy with leadership roles spanning 3 decades. We're lucky to have someone of his caliber focused entirely on accelerating therapies for Rett. Welcome, Bob.
Yeah, thanks a lot. And thanks also for assembling such a dynamic and productive team. It's really a pleasure to work with this group of people. So I'm going to introduce an approach which involves delivering a cargo which can correct a mutation in the genome, as Jenny described, and restore it to its wild type state and function. This is called DNA base editing. So this is done by introducing an enzyme which can combine with the small RNA as a targeting signal and correct a single base pair. This can be done virally or non-virally, and we're going to walk through that in a moment. The value of the approach lies in keeping the normal regulatory control of MECP2 expression and correction of the mutation without affecting expression of the wild type allele in mosaic girls. This removes risk for MECP2 overexpression, and if delivered efficiently, should be a one-time treatment without need for persistent expression of a replacement gene or RNA transcript. So very attractive features. The drawback lies in the allele by allele approach. It cannot be applied to all Rett patients, and it has a more complicated regulatory approach, which is really just being pioneered through the FDA as we speak. And this is called an umbrella trial, in which one clinical study can encompass multiple mutant alleles. So we could drop to the next slide. So we have really promising early results in mice and underway in non-human primates, which have demonstrated efficient allele correction and show a significant response to treatment. So it's really, really solid preclinical data that we've generated. And this has been done by our having assembled a consortium of investigators and funding a very mission-oriented approach with shared research results to build the opportunity. And that's what this slide is showing you. It's showing you the team. It includes Guoping Feng, a faculty member at MIT and director of the Rare Disease Nexus, Adrian Bird, who was introduced earlier, a distinguished Rett investigator from Edinburgh and on our board, and a trio of investigators from UMass Med School, John Watts, Eric Sontheimer, and Scott Wolf. We can drop to the next slide. So what are the delivery options for DNA-based editing, and how can we exploit these to build the most effective therapeutic? And so, as we mentioned, a viral delivery platform can be used to deliver base editing cargo, and with CapX, this can be an IV-infused treatment. The downside for viral delivery is that it maintains persistent expression of the base editing activity, which over time may lead to edits at unintended target sites in the genome, so toxicity risk. To avoid persistent expression, activity of the cargo can be regulated using a genetic circuit and a small molecule, which would be required to turn on activity. So there are methods we can use to regulate activity. The alternative solution lies in delivering activity in an mRNA format, as Jenny described, which by the nature of the RNA molecule will express activity and then decay. So this can be achieved by encapsulating mRNA in a lipid nanoparticle, which fuses with the cell membrane and delivers cargo. And this is essentially the Moderna COVID vaccine principle that we're all so familiar with. And so both of these are being very actively tested and pursued by RSRT. So here's some illustrations of non-viral delivery pathways. And so starting on the left, what we're showing you is a molecular structure of a ribonucleoprotein complex. This is the small targeting RNA guide coupled with the editing enzyme, which can be produced and purified and then delivered free in solution to the CSF. And so we've been involved with an investigator group at Yale who have shown feasibility of the delivery approach for Angelman's, and we hope to continue to see promising data during our research this year. In the middle panel, you see a complex called TERP, and it refers to a multi-layered lipid nanoparticle which can encapsulate an mRNA, so as I just described on the previous slide. So this is not unlike an oil and vinegar emulsion with fat droplets carrying a cargo. So here we're working with the biotech from Toronto who have shown that their particles can be delivered IV and cross the blood-brain barrier. So a very attractive delivery features. Again, the studies are at an early stage but hold great promise. Then the third path we're going down is to use exosomes. These are small particles which bud from human cells and can be manufactured by mammalian cell culture and can be engineered to contain the editing activity, so delivery vesicle, basically. And here we started a project with UK biotech, EVOX, to validate introduction of the particles into the CNF for neuronal editing. So you can see an extensive effort on our part to move away from AAV-based delivery and understand how we can exploit some of the emerging non-viral deliveries. Thank you. So I've tried to outline our efforts to correct Rett mutations in the genome and point out some of the pros and cons of the work. And it's really, as we stand now, what do we need to solve for? So question one, will the FDA endorse an umbrella trial approach for fixing multiple mutant alleles within one clinical trial? Like, very feasible. Beam Therapeutics was just approved to carry forward an umbrella trial approach in their studies on PKU. Second, can we approach this as a platform whereby a single enzyme can be combined with allele-specific guides? So working with a company called Profluent, trying to design an optimal editing platform. And then, as I just mentioned, can we solve for regulated or transient activity and avoid some of the safety concerns about persistent expression? So we're confident about finding solutions here and reaching our clinical trial submission target in late 2028, and we'll look at that pipeline again in a moment. So as Monica mentioned earlier, the genetic medicines are composed of two parts, delivery and cargo. And it's really the biochemical nature of the cargo that determine, and whether it requires persistent expression in the genome, that determine to a great degree the options for delivery. And so this figure is showing the delivery options for our modalities. Persistent expression requirements keep gene supplementation and RNA trans-splicing locked into viral delivery. Base editing can be delivered either virally or non-virally, with non-viral delivery providing the best option for controlled activity and repeat dosing. And finally, with ADAR-based RNA editing, this is envisioned as a non-viral delivery platform, which reflects a routine delivery schedule of these small RNAs to the CSF, but it also has options for using viral delivery and therefore only requiring one treatment. So on my final slide, I wanted to summarize where we are with these modalities and press home with this chart how the nature of the Rett mutation in individual girls lines up with treatment options, with the primary distinction being whether or not the approach is done in an allele by allele fashion or whether one therapeutic covers all mutations. So please, drop back to this after the webinar and understand, where your child falls, and we're happy to provide other information about the modality and stage of development. So Monica, thank you, and back over to you.
Thank you, Bob. And if you're unsure about what mutation your child has and what category here it fits in, feel free to email me. And I will help you out, and I can help you understand your child's genetic mutation if you don't quite understand it yet. Okay, so now that you have all that context, and I know it was a lot of information, but we'll give you a recording. You can listen to this all again if that helps. I'm going to bring you back to our goal, right? To have applications requesting approval to initiate clinical trials submitted to regulators by 2028 for 3 novel genetic medicines. And I think the slide, speaks for itself. But what you see here in terms of the timeline is our best estimate given current data. What I will add is that RSRT's involvement with these programs will be data-driven. We'll provide support if the data warrants it and continue and increase our support, and we'll pivot if and when the data suggests that we need to do that. I cannot promise that these timelines will pan out, but I can promise you that our partners and everyone at RSRT will do our very best to adhere to them. Now, our roadmap to cure goal was to have 3 novel programs, and we'll let the data determine which 3. And if the data looks good for all of the programs, and if we have the financial resources, we'll advance all 4, right? The more, the better. Now, beyond the companies that we've already discussed, there are additional startups and academic labs that are pursuing these programs. And the programs that we are contributing funding to are bolded. Now, in the gene supplementation space, as you can see, it's getting pretty active, right? So beyond Neurogene and Taysha and Reverta that we introduced to you today, there's also Emugen. Emugen is a startup based in Boston that is also pursuing MECP2 gene therapy with CapX. Additionally, there is an academic-sponsored clinical trial using AAV9 that is currently active and enrolling in China, and another Chinese trial starting in the next year or so that is using a capsid that I would describe as being somewhere in between AAV9 and CapX. There is also another gene supplementation program that has not yet been publicly disclosed. In the RNA editing space, we're funding a project with Shape Therapeutic that uses a novel viral vector that can also get into the brain. Wave and Vico also have Rett programs on their pipeline, although there hasn't been any recent updates from the companies. RSRT is also funding 2 startups in the trans-splicing space beyond Tacit. One is Transit, one is a newco still in stealth mode, and then the NIH is funding a base editing program. And I've got to say that it's just wonderful to see all this activity. I remember a time when obviously that wasn't the case. Okay, so I explained earlier the steps that are required in getting a lab discovery into the hands of patients. And historically, RSRT has funded academic labs who make a discovery, the university files intellectual property, and then biotech companies license it and develop the drug. This is what we've seen in the case of Neurogene and Taysha, for example. And the big advantage is that companies bring capital and they bring expertise required to move the programs forward. But the downside is that RSRT loses control of the programs. And again, I'll bring up the Novartis Rett program, which got its start at RSRT, but then once licensed to Novartis was terminated. Now, it's unlikely that RSRT will have sufficient funds to bring the program all the way from discovery to commercialization. However, the longer we can keep a program internal, or the more funds that we can contribute to a Rett program within a company, for example, Tacit or Reverta, the more influence we have. Now, here are some examples of why having control is desirable. With financial resources, we have control of experimental designs and rigor, timelines and milestones. We would control the clinical trial design, for example, whether to include males in clinical trials. Importantly, we would have access to the data, which is critical. And learnings from one program would inform other programs. For example, learnings from manufacturing CapX for a gene supplementation program would give us valuable information about a trans-splicing program that also uses CapX. Now, at some point, we would certainly need biotech partners, but the point is to hold on to the programs for as long as possible. So how much does it cost, roughly speaking, to bring a genetic medicine program from the lab through a clinical trial of 15 people? We're not talking about all the way to commercialization, just the first clinical trial. As you can see here, it takes roughly $30 million. Now, this is a bare bone budget, which reflects the lean way in which RSRT operates. The required work would be done at contract research organizations, so we take full advantage of a virtual environment with no cost going to infrastructure. So advancing 4 genetic modalities, 4 genetic medicines would take about $120 million. Now, I'm going to turn things over to my colleague, Tim Freeman, who's our Chief Development Officer. It has been my pleasure to have Tim as a colleague for the last 13 years. And as you can imagine, our mission is very close to his heart.
Thank you, Monica. Hi, everyone. It's so nice to be here with so many fellow Rett families. Since RSRT was founded in 2008, more than $115 million has been contributed to the organization. So that's averaging about 6.5 million raised a year. In the last 4 years, we've held steady at around 8 million raised every year. And before that, for several years running, we were raising even more than that. So who is raising these funds for RSRT, and where did those funds come from? I'm going to give you a couple of facts. Exactly 0 of those funds, $0, have come from the government. And you may be a bit surprised to know that less than 5% of that funding has come from corporations, and it's not unique to RSRT. Corporations are a small percentage of philanthropic giving across the board to all causes. So the vast majority of our funding, over 90%, has come from individual people. And very, very importantly, 100% of the donations made to RSRT over the last 18 years have come from Rett families and their networks. So that means when a Rett family takes action and asks their network to make a donation, donations come in. So we are enormously, enormously grateful to the families in the US and around the world that have contributed and fundraised. They are why RSRT has made this exciting progress that you're hearing about. Those of you already fundraising and giving are fueling this research at RSRT. So of the approximately 350,000 Rett families around the world, I want to ask you, how many do you think are fundraising and contributing to RSRT? And of the approximately 15,000 families in the US, how many are fundraising and giving? So we're going to launch 2 poll questions here now that give you a chance to give your best estimate on those 2 questions. So what percentage of families in the US are fundraising or donating to RSRT, and what percentage of families around the world do you estimate are fundraising or donating for RSRT? And I hope you'll all participate in this. I think it'll be interesting. And we'll give you a minute or so to respond here. Okay, we have responses in.
All right, let me end it. Last chance. If you haven't responded yet. Okay, here we go. I'm going to end it. Share results.
Okay, so interesting. It's really everyone thought in US families sort of approximately the same for each level. 28% of you felt like 3% were giving, and 28%, 15%, and 23% and 21% for the higher percentage of families giving. And 37% felt like 0.1% were giving globally, and 23% felt like 5%, and then 25%, 15%. Interesting. Okay. So thank you for participating in that. So we're going to give you the actual percentages here.
Oh, it's not. Hold on. It's not. Here we go.
There we go. So the actual percentages are 3% of US families have fundraised or contributed to RSRT, and 0.1% of global families have fundraised or contributed to RSRT. So that's 1/10 of 1% of the entire Rett family population. I do want to make a point here regarding families outside the US. The numbers are lower there in part because there are countries that have their own Rett organizations that Rett families in those countries fundraise for. So most of those organizations focus on family support. Some do fund some basic research. However, they are not funding genetic medicine programs with the goal of getting to clinical trials as RSRT is. So my purpose behind sharing this information is to let you all know what huge potential there is to raise more funds and that the power to push this research ahead lies with you and only you, Rett families. So imagine what we could accomplish and how much faster we would move forward if we increased the percentage of Rett families that fundraised in the US from 3% to, say, 10%. Or imagine what we could achieve if we increased global Rett family fundraising for RSRT from 0.1% to 3%. So that brings me to the all-important question of what can you do? And I hope you'll all reach out to me. First of all, I love talking with other families, and I love brainstorming and thinking about this stuff. So specifically, there are different ways to fundraise, and I hope you'll choose one that feels comfortable to you. So number one, you can join an existing event. We have about 20 major events a year around the country. And if you join a committee for one of those events, really all the work is done for you. All you need to do is invite your network. And we have events in LA and New York City and New Jersey and Philadelphia, Austin, Texas, Chicago, Pittsburgh, Washington, D.C., Phoenix, Seattle, Michigan, and more. So if you contact me, I can connect you with the family running an event, and you can get started right away. Number two, you can start your own event. An event can really be anything you envision. It does not have to be a gala. It can be a backyard picnic or barbecue. It can be a walk. It can be a happy hour at a restaurant or a bar. Last weekend, we even had an event that was a car show. So again, contact me, and we can come up with something that would be fun for your network and for you. Number three, you can start a crowdfunding campaign. So we use GoFundMe as our crowdfunding platform. This is super easy to do. You can personalize your campaign with photos and a message. And we've had crowdfunding campaigns that have raised $500, and we've had crowdfunding campaigns that have raised $100,000. And we are grateful and proud of all of them. What matters is that you give it a try. A fourth way you can fundraise is you can start a purple lemonade stand, and you'll hear more about this soon from RSRT. This is a fun and easy way to raise funds in your neighborhood. We did this initiative last summer, and we're going to do it again this year. So stand by to hear more about that. Number five, you can start a letter or email campaign. And I know this sounds a little low tech and old fashioned, but it works. I've done this with my own network and gotten good results. And again, contact me. I am more than happy to help you craft your message. And number six, ask about employee matching. So if you work for a company, go to your HR office and ask about matching gifts, or sometimes bigger companies will have a philanthropic branch or a foundation. Talk to someone there about the company making a donation to RSRT. Contact me, and I can help you with an approach. So this is really big, actually. A month ago we launched a matching campaign. A family in Pittsburgh very generously is matching dollar for dollar every donation made on this campaign and to crowdfunding campaigns that families set up. But they're only doing this through July 1st. So we've got a week to go to start your own crowdfunding campaign and have your donations doubled. So I really hope a lot of you will consider taking advantage of that right now. And the message I'll leave you with is I am always here to help you with any of this, to talk about it with any of you. So please, please, please contact me anytime. Thank you so much.
Great. Thank you so much, Tim. And I just also want to echo Tim's sentiments. We're so grateful for everyone that supported RSRT. And if you haven't, you want to give it a try. I hope that I've convinced you that we're worthy of your support. And, you know, we want to work together to end Rett syndrome. Okay, I am going to stop sharing. We are going to answer your questions. I was so focused on the webinar that I haven't looked at the questions that have come in yet. So I'm going to just kind of scroll through them quickly. Okay, so one question that I know we're going to get a lot of, and I see a couple already, and I'm going to hand this over to Jenny to answer, but it's basically, where are the clinical trials going to happen for these novel genetic medicines? If Reverta, is an Italian company, does that mean, or Procure, which is from the Netherlands, does that mean the trials will be there? So Jenny, can you talk to that?
Yep. Happy to answer that question. There's no necessary connection between where a company is based, like Italy or the Netherlands, and where the clinical trials sites are that actually are conducting the trial. Nor does it matter. So it really is driven more by the expertise of a particular investigator, a doctor in a particular hospital or institution. It's important that they understand Rett syndrome, but also that they have experience, running clinical trials, and also experience with genetic medicines. And so that's a very specialized niche, and a lot of that governs where trials will be conducted for genetic medicines. But as the field has grown, there are many, many more sites and locations that have the requisite expertise in the United States, in Europe, in Australia, China, etc. So the options are ever growing, and it's kind of a strategic decision about where you want to go first to be most efficient and to learn the most you can about your investigational therapy quickly and safely.
Great, thank you, Jenny. I'll quickly answer a couple of these. If one gets dosed using AAV9, can one be redosed using CapX? Unfortunately not. Although CapX is an engineered capsid, it's still part of the same family. And so they're closely related enough that if you get AAV9, you cannot get CapX. Would the Italian startup seek approval in the EU or in the US? I think the goal will be both. I mean, both Europe and US are huge markets for companies. They really can't afford to ignore one or the other. So they will attempt to do both. We have an MECP2 deletion. Would editing be our route? So your route will be gene supplementation, RNA trans-splicing, eventually when we bring back prime editing, and MECP2 reactivation. But the editing approaches would not work. Let's see. There was one interesting question here. I haven't. Okay, and I'm going to leave it up to Bob, John, or Jenny to see who wants to answer this. Curious about viral shedding with AAV9. If an individual receives AAV9 therapy, such as from neurogene or Taysha, can they shed that virus and cause others to contract it and develop an immune response against it?
Mm-hmm.
You want to answer it, Jenny?
Yeah, sure. That can happen. And there's actually guidance from FDA about how to track that over time. They have requirements of collecting urine samples, stool samples, saliva samples after dosing to follow it. The amounts will go down basically as the body has cleared out the virus over time. But there is a chance for exposure to family members, etc. The good news is that AAV doesn't cause illness. It means that, your body sees it and you make an antibody against it, you mount an immune response, but it's not going to make anybody sick per se. The one issue is for example, in the Duchenne world, if there are two brothers who have Duchenne and they're not dosed at the same time, there's a chance of the first dose brother exposing the younger dose brother, to be dosed brother. Such that he now has anti-AAV9 antibodies. And there's ways around that, but that's the main consideration. It wouldn't be such an issue, I think, in the context of Rett syndrome.
Jenny, I think we're going to keep you busy here because there's a lot of clinically oriented questions. Yep. With the RNA trans-splicing, what age would you start with for trials? I'm going to
That's
start by
also-
saying we don't know yet.
Yeah,
Nothing's been established, but go ahead. Jenny.
Yeah, that's the bottom line. Nothing's been established yet. Really, for any of these trials, looking for both safety and effectiveness of an investigational new genetic medicine, what's most helpful is to understand kind of the population, the age group in which you're most likely to see a change, an effect. And so, for example, early on, when skills are waxing and waning in patients who are kind of going through that phase of gaining skills and then maybe losing some, you never know what direction they're going to go. But at some point, they reach a plateau in which nothing's changing, no skills being lost or being gained. So there, if anything changes, you're more likely to be able to attribute that to an effect of the drug, as opposed to just the fluctuations that happen with the disease itself. And that's really the approach that both Neurogene and Taysha have been taking. There's a big body of natural history data that was collected over many years that have been used to kind of look at the shape of the disease over time. And after age 6 or so, there's a single-digit percentage of gaining a new skill. And so at that point, any new skill that raises its head above that background can be attributed to the drug. And that's actually what they've negotiated with the FDA about having a responder and having a successful trial for approval. Are they starting to see milestones being achieved during that time that otherwise milestones would have been flat?
I just
Great,
want to
thank
add
you.
one thing. There is also a difference between a clinical trial design and a label on an approved drug. So we're going to get a lot of questions, I think, on trials, but there's not always a one-to-one concordance. Is that correct, Jenny?
It depends, and there's kind of an art to that in the regulatory negotiations and how you design your trials and what populations, what groups of patients that you include in your studies. The idea, obviously, is to have as broad and non-specific, if you will, a label as possible. The label says who you can treat, what the drug is thought to be able to do, what the safety issues are, how you make it, how you give it. But, for example, payers might not pay if it's for a group of patients that weren't specified on the label. So actually having that population be as broad as possible, for example, not specifying girls or boys, for example, not specifying an age or stage of disease, those kinds of things. And that's something that all programs strive for, to be able to have a broad label that will let the most possible patients get access to the new drug. But
Great.
that needs to be negotiated with FDA.
Here's a question for John. RNA, and then I'm going to have one for you, Bob. RNA is temporary, unlike DNA. So editing RNA will last for what period of time? Can RNA be done multiple times?
Yes. So, excuse me. RNA is temporary. That's absolutely true. RNA editing, I'm going to start with, which is where you're making these single base corrections. So because that is relying on these ADAR enzymes, you can dose the guide either once with a virus like CapX, or you can dose multiple times. Viruses are for permanent expression, so they're typically one and done. Non-viral approaches like dosing an antisense oligo are done but degraded. And so typically it's about 3 to 4 times a year that you need to dose an oligo. There's not the same immune concern over oligonucleotides as there is with viruses. Now, to switch over to trans-splicing, because you're moving from editing an individual base to something much, much larger, there simply aren't oligonucleotides that you can manufacture that are big enough to perform trans-splicing using a transient delivery system. And so for trans-splicing, you would want to use a virus like CapX.
Great, thank you. Bob, let me just find that question. To what extent is AI currently being used to accelerate the development of therapies for Rett, and do you expect future advances in AI to further shorten the timeline for bringing such treatments to patients?
And I think that the two dominant pathways that are being used are in the design of proteins, the complexes that are used to achieve base editing or achieve some of the gene supplementation work. That's also being applied to design of RNA molecules so they have the appropriate structure and work with the greatest potency. And so I think we mentioned earlier, we're working with a group called Profluent that's doing exactly that to build out our portfolio of base editing enzyme complexes. You know, Jenny may comment in a sec, but the other way AI is becoming used dominantly is in clinical trial data review and in clinical trial design. So let me drop that over to you, Jenny, just for a comment. And
Oh, you're muted, Jenny.
Darn it, I just got to leave it unmuted. So yeah, absolutely true, Bob. That's increasingly being done. For clinical trial design, are there kinds of endpoints that will be the most informative, and you could apply AI analyses to a big body of natural history data, for example. Many ways that you can apply AI to make the clinical trials more efficient, to learn more earlier in ways that you might even be able to modify your approach for a later part of the trial or a later study. And so it's very much being applied to make the process more rapid, more efficient, fewer patients exposed, less time, less cost, which benefits everybody in the long run. So
we're getting a lot of questions about, if we've had virally delivered treatment once, can we get something that's non-viral? Yes, the answer is yes. So it's just that you can't get, for now, viral and viral, right? You get viral and non-viral. The non-virals for now are base editing and RNA editing, and that's a subset of mutations. It's a pretty high subset. It's up to 40, 50%, but it's not everyone. The other thing I want to mention is there are efforts underway at companies trying to figure out how to make these viral vectors go into like stealth mode so that they're undetected by the immune system. And I haven't heard updates recently, but hopefully in time, that's something that they'll figure out. And then, this whole one bite at the apple issue will go away, and that will make all of us feel a lot better.
I do want to build on something there, Monica, and that is, if you have received a treatment, you will very, very likely be ineligible for a clinical trial for a second type of medicine. So if you've gotten an AAV gene therapy, you're very unlikely to be able to be enrolled in, say, an RNA editing. That doesn't mean that you would be precluded from getting an approved treatment. But when you're designing a clinical trial, you don't want confounds of this patient has already had a therapy.
Well, here's an interesting question.
Could I comment a little bit about the
Jenny.
prospect of redosing? So there's a lot, as you can imagine, there's a tremendous interest within the field about how to get around this, to permit, redosing, just standard AAV approach to gene delivery. There's different ways that this can be tackled. One is to just give a whole lot of immunosuppression around the time that the AAV is given, so the immune system doesn't see it, so to speak, and then doesn't mount that memory response, so that you can redose later. That's one approach. Another approach people have looked at is to use an actual enzyme. I think it's from bacteria, but that will cut up antibodies. So the body may make antibodies, but they'll be destroyed. And so that antibodies are part of what enables the memory response to happen later. So if that never happens, then you're better off and may not have a memory response later with re-exposure. So there's lots of ways that are being looked at to get around that, to now allow redosing to be routine, as opposed to impossible right now.
Okay. Here's a good question. How can the FDA's plausible mechanism of action speed up development of cures for all genetic mutations on the MECP2 gene? So the FDA has been coming up with some, new mechanisms. Jenny, do you want to start that? And
sure. And I know John has thought about this a lot and is a real kind of proponent of the excitement around this initiative. So, John, please feel free to leap in.
You're muted, John.
So yes, the FDA has been looking at new ways to make it easier to develop genetic medicines. And one of those approaches is called plausible mechanism. And largely that has been something that hasn't been utilized yet. It was a proposal and it was written up in an article, but the tangible methods for how to use plausible mechanism for an approval, to my knowledge, at least, have not been outlined where you have a clear path on how to do that. I think that there are ways that we can take advantage of pathways such as plausible mechanism. And late last year, as an organization, we, with some of our collaborators, went through some exercises on how could we do that and what programs could be amenable and how could we push the FDA to try and make novel pathways on how to get drugs into the clinic and approved faster. I'm not sure that answered the question exactly. There's a lot of details in plausible mechanism that, but at the moment, it's not something that I feel like we could design a clinical trial around today.
So here's a question. When do you think RSRT will be completely out of business and Rett functionally cured? Well, not fast enough.
Yeah.
I wish, I've been doing this 28 years, 27 years. It's hard to answer that question. And I really don't want to give a timeline and then, find out that it wasn't accurate. I can tell you that first of all, that is our goal. We want to be out of business. We are not a nonprofit that wants to continue ad nauseum. And, I'm hoping that we'll have a better idea of an answer to that in the next, 5 years. It does take time to go through all the steps that I showed in the earlier, right? When we started all those steps 1 through whatever it was, 9, it takes time. And then once the clinical trials are done and the data has to be analyzed and the regulators have to go through it and they have to decide, and then it takes time. But I'm optimistic. And also, I also want to mention, cure. Like, do I expect my daughter at age 30 to have a cure? No, I absolutely do not. And I think a lot of parents are very realistic when it comes to that. We are not promising that all of your children are going to be cured. Cure is our aspirational goal, and that's what we're working towards, and with incredible optimism and urgency and passion. Whether we can cure a 10-year-old, a 5-year-old, a 2-year-old, or a newborn remains to be seen. Our goal is a cure, but there's also so much room, and I'll use my daughter as an example, between where she is right now and a cure, right? A lot of space in there. And I will take improvements, not minimal improvements. I want dramatic improvements. But I know that a cure is not possible at age 30, but I'm holding out for dramatic improvements. And I think that's possible. And the animal data suggests that I'm not being, pie in the sky, Pollyanna-ish about it. So that's what I can say. And yeah.
I'd also like to just add that what RSRT has done is becoming the model for what other patient organizations are doing in genetic medicines. I've been to many conferences and they'll say, we look at what RSRT did with their preclinical, with their mouse models, with their biorepositories, and other patient organizations come to us and say, what have you learned? Can we follow in your footsteps to accelerate things? And it's never, ever, ever going to be fast enough. Yeah. But because of the efforts of Monica and what you have enabled, it's moved much, much faster than it normally would have.
Yep. Here's a question. How are the mutations selected for RNA editing? Are some easier to edit than others?
So, yes. So the enzymes that are responsible for this, the ADAR enzymes, have a normal preference. They edit a single base, but they care who the neighbors are. And so there are some that are easier to edit than others. One of the other pieces I think that's important, and this is getting into some of the details, is for G to A mutations, we can correct back to the normal sequence. And for the nonsense mutations like 168, 255, 270, 294, what the RNA editing is actually doing is removing that X, and that X is a stop. And so basically the cell is making the protein and it comes up to this truncation and says, nope, no more. And so RNA editing is limited because it can only make one type of change. And so we can't change that back to the normal sequence, but we can allow it to keep reading through and get to the rest of the functional domains of MECP2. And in efforts by Gail's lab, by other labs in the editing consortium, and really by RSRT, we've been able to show that that's enough. That is therapeutic, even if it's not going back to the true normal sequence. You can use RNA editing to allow for full-length MECP2 expression.
Okay, last question. And then we'll try to answer the rest of the questions. We'll send an email, or we'll figure out a way to address the rest of the questions. But this is a question that I think probably lots of you have, which is why I'm selecting it. Once treatments become commercialized, what would the time frame be for the public being able to access these? We're based in New Zealand. I'll just get it started, and then I'll turn it over to Jenny, I think, or anyone else who wants to jump in. So the way my mind works is always like, let's tackle one big problem at a time. And, first it was, we have to get basic science going. And then it's like, let's get genetic medicine programs going. And now companies are involved. And first we have to have something that really works, right? And then the companies will be play a very pivotal role in applying for regulatory approval, hopefully across the world. Then we got to get payers to pay it. So to pay it, because these are going to be expensive medications, and most families, including mine, are not going to be able to pay out of pocket for this. So Jenny, how long do you think all that takes?
Oh,
You're on the hot seat now.
I'd be hard pressed to put a time frame on it. And I really do think it depends on what particular country or geography one is in. For example, in some countries, at least this used to be the case. For example, once FDA has approved a particular new therapy for the United States, there are some countries that will kind of almost take that as a rubber stamp for their approval in their country. But other countries have their own standards. They want to go through their own process, and that would require another kind of regulatory review and the time that would take. And the payment structures also differ tremendously across countries and regions. Sometimes it's very centralized, sometimes it's very kind of fragmented, as it is in the United States, although, probably families with Rett, dealing with Rett syndrome, they may have more centralized kinds of approaches to payment for medical care. So there's no good one good answer, I'm sorry. There's a lot of factors that play into it. But one of the things that advocacy organizations can do is to kind of engage with local providers, have clinical sites in a country where you want to be able to get access to patients there, kind of advocate for the need with health authorities in the respective countries. So there are ways to kind of lobby to make that process more efficient, more quick, to demonstrate the urgency of the need. And that can actually help move the needle, I think.
Thank you, Jenny.
Mm-hmm.
All right. Thank you to everyone for giving us an hour and a half of your time. I know everyone is busy. Thank you to my colleagues for every day. It's a joy to work with all of you. And I'm
Likewise.
Yeah, I'm not going to get emotional, but we're doing really important work, and to be able to do it with people that you respect and really enjoy is a blessing. We are going to send you all a recording so you can listen to this again. And please send us your questions. And if you're interested in helping to support us, please get in touch with Tim. All right. Thank you. And the other thing is we want to do a lot more webinars. So on particular topics. So that's the goal. If you don't hear from us, sometimes you think nothing's going on. It's actually the opposite. We're busy.
Too busy.
But we do want to really try to be better at keeping you updated. All right. Thank you, everyone. Bye-bye.
Thanks, everybody.